Hello dear students. Today we are going to read our new chapter. First of all, we'll do reading and then we'll move forward. Chapter 8, How do organisms reproduce? Before we discuss the mechanism by which organisms reproduce, let us ask a more basic question. Why do organisms reproduce? After all, reproduction is not necessary to maintain the life of an individual organism. Unlike the essential life processes such as nutrition, respiration, or excretion, on the other hand, if an individual organism is going to create more individuals, a lot of its energy will be spent in the process. So, why should an individual organism waste energy on a process it does not need to stay alive? It would be interesting to discuss the possible answer in the classroom. Whatever the answer to this question, it is obvious that we notice organisms because they reproduce. If there were to be only one non-reproducing member of a particular kind, it is doubtful that we would have noticed its existence. It is the large number of organisms belonging to a single species that brings them to our notice. How do we know that two different individual organisms belong to the same species? Usually, we say this because they look similar to each other. Thus, reproducing organisms create new individuals that look very much like themselves. Let's see next 8.1. Do organisms create exact copies of themselves? Organisms look similar because their body design are similar. If body designs are to be similar, the blueprint for these designs should be similar. Thus, reproduction at its most basic level will involve making copies of the blueprints of body design. In class 9, we learned that the chromosome in the nucleus of a cell contain information for inheritance of features from parent to next generation in the form of DNA, deoxyribonucleic acid molecule. The DNA in the cell is the information source for making protein. If the information is changed, different protein will be made. Different protein will eventually lead to altered body design. Therefore, a basic event in reproduction is the creation of DNA copy. Cells use chemical reactions to build copies of their DNA. This creates two copies of DNA in a reproducing cell, and they will need to be separated from each other. However, keeping one copy of DNA in the original cell and simply pushing the other one out would not work, because the copy pushed out would not have any organized cellular structure for maintaining life process. Therefore, DNA copying is accompanied by creation of an additional cellular apparatus, and then the DNA copy is separated, each with its own cellular apparatus. Effectively, a cell divides to give rise to two cells. These two cells are of course similar, but are they likely to be absolutely identical? The answer to this question will depend on how accurately the copying reaction involved occur. No biochemical reactions is absolutely reliable. Therefore, it is only to be expected that the process of copying the DNA will have some variations each time. As a result, the DNA copies generated will be similar but may not be identical to the original. Some of these variations might be so drastic that the new DNA copy cannot work with the cellular apparatus it inherits. Such newborn cell will simply die. On the other hand, there could still be many other variations in DNA copies that would not lead to such drastic outcome. Thus, the surviving cells are similar to but subtly different from each other. The inbuilt tendency for variations during reproduction is the basis for evolution, as we will discuss in next chapter. The importance of variations we have to see now in this topic. Population of organisms fill well-defined places or niches in the ecosystem, using their ability to reproduce. The consistency of DNA copying during reproduction is important for the maintenance of body design features that allows the organism to use that particular niche. Reproduction is therefore linked to the stability of population of a species. However, niches can change because of reasons beyond the control of an organism. Temperatures on Earth can go up or down. Water level can vary, or there could be meteorite hits. To think of a new example, if a population of reproducing organisms were suited to a particular niche, and if the niche were drastically altered, the population could be wiped out. However, if some variations were to be present in a few individuals in this population, there would be some changes for them to survive. Thus, if there were a population of bacteria living in temperate waters, and if the water temperature were to be increased by global warming, most of these bacteria would die, but the few variants resistant to heat would survive and grow further. Variation is thus useful for the survival of species over time. Some in-text questions are given here are: what is the importance of DNA copying in reproduction? And second question is: why is variation beneficial to the species but not necessary for individual? Next point, mode of reproduction used by single organism. One activity is given here. Activity 8.1: dissolve about 10 gram of sugar in 100 ml of water. Take 20 ml of solution in test tube and add a pinch of yeast granules in it. Put a cotton plug on the mouth of the test tube and keep it in a warm place. After 1 or 2 hours, put a small drop of yeast culture from the test tube on a side and cover it with cover slip. Observe the slide under microscope. Activity 8.2 is given. Wet a slice of bread and keep it in a cool, moist and dark place. Observe the surface of the slice with magnifying glass. Record your observation for a week. Compare the contrast, the way in which yeast grows in the first case and how mold grows in the second. Having discussed the context in which reproductive processes work, let us now examine how different organisms actually reproduce. The mode by which various organisms reproduce depend on the body design of the organism. First process is fission. For unicellular organisms, cell division or fission leads to the creation of new individual. Many different patterns of fission have been observed. Many bacteria and protozoa simply split into two equal halves during cell division. In organisms such as amoeba, the splitting of the two cells during division can take place in any plane. Binary fission in amoeba is shown here. However, some unicellular organisms show somewhat more organization of their bodies, such as seen in Leishmania, which causes Kala Azar, we have seen in 9th standard, which have a whip-like structure at one end of the cell. In such organisms, binary fission occurs in a definite orientation in relation to these structures. Other single-cell organisms, such as malarial parasite, Plasmodium, divide into many daughter cells simultaneously by multiple fission. Yeast, on the other hand, can put out small bud that separate and grow further, as we see in activity 8.1. So next part is fragmentation. Here one activity is given. Here multiple fission in Plasmodium is shown. One cell and many daughter nuclei are formed. Afterwards that cell will break. Activity 8.4: collect water from a lake or a pond that appears dark green and contain filamentous structures. Put one or two filaments on slide, put a drop of glycerin on these filaments and cover it with cover slip. Observe the slide under microscope. Can you identify different hues in the Spirogyra filament? In multiple cellular organism with relatively simple body organization, simple reproductive method can still work. Spirogyra, for example, simply break up into smaller pieces upon maturation. These pieces or fragments grow into new individual. Can we work out the reason for this based on what we saw in activity 8.4? This is not true for all the multicellular organisms. They cannot simply divide cell by cell. The reason is that many multicellular organisms, as we have seen, are not simply a random collection of cells. Specialized cells are organized as tissues, and tissues are organized into organs, which then have to be placed at definite positions in the body. In such a carefully organized situation, cell by cell division would be impractical. Multicellular organisms therefore need to use more complex ways of reproduction. A basic strategy used in multicellular organisms is that different cell types perform different specialized functions following this general pattern. Reproduction in such organism is also the function of specific cell type. How is reproduction to be achieved from a single cell type? If the organism itself consists of many cell types, the answer is that there must be a single cell type in the organism that is capable of growing, proliferation, and making other cell types under the right circumstances. Regeneration. Many fully differentiated organisms have the ability to give rise to new individual organism from their body parts. That is, if the individual is somehow cut or broken up into many pieces, many of these pieces grow into separate individuals. For example, simple animals like Hydra and Planaria can be cut into any number of pieces, and each piece grows into complete organism. This is known as regeneration. Regeneration is carried out by specialized cells. These cells proliferate and make large number of cells from this mass of cells. Different cells undergo changes to become various cell types and tissues. These changes take place in an organized sequence referred to as development. However, regeneration is not the same as reproduction, since most organisms would not normally depend on being cut up to be able to reproduce. So here in this figure, regeneration in planaria is shown. The planaria is cut into 3 parts and each part is growing as a new individual. Next is budding. Organisms such as Hydra use regenerative cells for reproduction in the process of budding. In Hydra, a bud develops as an outgrowth due to repeated cell division at one specific site. These buds develop into tiny individuals and when fully mature, detach from the parent body and become new dependent individual. You can see here budding in Hydra. Hydra body is shown when bud formation takes place and then bud get detached from the parental body and will grow as a new individual. Next part is vegetative propagation. There are many plants in which parts like the roots, stem and leaves develop into new plants under appropriate conditions. Unlike in most animals, plants can indeed use such a mode of reproduction. This property of vegetative reproduction is used in methods such as layering or grafting to grow many plants like sugarcane or roses or grapes for agricultural purpose. Plants raised by vegetative propagation can bear flower and fruit earlier than those produced from seeds. Such methods also make possible the propagation of plants such as banana, orange, rose and jasmine. that have lost the capacity to produce seeds. Another advantage of vegetative propagation is that all plants produced are genetically similar enough to the parental plant to have all its characteristics. Here, activity 8.5 is discussed. Take a potato and observe its surface. Can notches be seen? Cut the potato into small pieces such that some pieces contain a notch or bud and some do not. Spread some cotton on a tray and weight it. Place the potato pieces on this cotton. Note where the pieces with the buds are placed. Observe changes taking place in these potato pieces over the next few days. Make sure that the cotton is kept moistened. Which are the potato pieces that give rise to fresh green shoots and roots? That we have to observe. Okay. Similarly, buds produced in the notches along the leaf margin of bryophyllum fall on the soil and develop into new plant. Here, leaf of bryophyllum with buds is shown. This plant is generally available at our doorsteps. Bryophyllum, it is in Marathi, it is called as a पानफुटी, commonly it is known as a पानफुटी, because उसका एक पत्ता अगर गिर गया तो उस पत्ते से नया प्लांट आ जाता है, नए प्लांट से आ जाते हैं. तो this plant you might have observed at many places. Activity 8.6, select a money plant. Cut some pieces such that they contain at least one leaf. Cut out some other portion between two leaves. Dip one end of all pieces in water and observe over the next few days which one grows and give rise to fresh leaves. What can we conclude from your observations? So this is one simple activity for all of us. Money plant is available at each doorstep. Everybody is having money plant. So what you have to do is you have to cut its stem into different parts and put in bottle. Simple bottles or mineral water bottles also can be used. You have to fill up this bottle with water and you have to keep that plant in the water and observe. Next is tissue culture. In tissue culture, new plants are grown by removing tissues or separating cells from the growing tip of plant. The cells are then placed in an artificial medium where they divide rapidly to form a small group of cells or callus. The callus is transferred to another medium containing hormones for growth and differentiation. The plantlets are then placed in the soil so that they can grow into mature plants. Using tissue culture, many plants can be grown from one parent in disease-free condition. This technique is commonly used for ornamental plants. Nowadays, it is used for banana, sugarcane, for pomegranates, for grapes. It's become generalized now. Next is spore formation. Even in many simple multicellular organisms, specific reproductive part can be identified. The thread-like structure that develop on the braid in activity 8.2 above are the hyphae of the braid mold, that is rhizopus. They are not reproductive parts. On the other hand, the tiny blob on a stick structure are involved in reproduction. The blob are sporangia, which contain cells or spores that can eventually develop into new rhizopus individuals. The spores are covered by thick wall that protect them until they come into contact with another moist place and can begin to grow. Here, spore formation in rhizopus is shown. All the modes of reproduction that we have discussed so far allow new generations to be created from a single individual. This is known as asexual type of reproduction. Some in-text questions are given here. Let us discuss about it. How does binary fission differ from multiple fission? How will an organism be benefited if it reproduces through spores? Can you think of reasons why more complex organisms cannot give rise to new individuals through regeneration? Why is vegetative propagation practiced for growing some type of plants. Why DNA copying an essential part of the process of reproduction? So these are in-text questions. Later on we are going to discuss. Next part is sexual reproduction. We are also familiar with modes of reproduction that depend on involvement of two individuals before a new generation can be created. Bulls alone cannot produce new cows, nor can hens alone produce new chicks. In such cases, both sexes, males and females, are needed to produce new generations. What is the significance of this sexual mode of reproduction? Are there any limitations of the asexual mode of reproduction, which we have been discussing above? Why the sexual mode of reproduction? The creation of two new cells from one individual involves copying of DNA as well as of the cellular apparatus. The DNA copying mechanism, as we have noted, cannot be absolutely accurate, and the resultant errors are a source of variation in populations of organisms. Every individual organism cannot be protected by variations, but in a population, variations are useful for ensuring the survival of the species. It would therefore make sense if organisms came up with reproductive modes that allows more and more variations to be generated. While DNA copying mechanisms are not absolutely accurate, they are precise enough to make the generations of variations a fairly slow process. If the DNA copying mechanism were to be less accurate, many of the resultant DNA copies would not be able to work with the cellular apparatus and would die. So, how can the process of making variants be speeded up? Each new variation is made in a DNA copy that already has variations accumulated from previous generation. Thus, two different individuals in a population would have quite different patterns of accumulate variations. Since all these variations are in living individuals, it is assured that they do not have any really bad effects. Combining variations from two or more individuals would thus create new combinations of variations. Each combination would be novel since it would involve two different individuals. The sexual mode of reproduction incorporates such a process of combining DNA from two different individuals during reproduction. But this creates a major difficulty. If each new generation is to be the combination of the DNA copies from two pre-existing individuals, then each new generation will end up having twice the amount of DNA that the previous generation had. This is likely to mess up the control of the cellular apparatus by the DNA. Besides, if generation doubles its DNA, fairly soon there will be the only DNA on Earth, with no room for anything else. How many ways can we think of for solving this difficulty? We have seen earlier that as organisms become more complex, the specialization of tissue increases. One solution that many multicellular organisms have found for the problem mentioned above is to have special lineages of cells in specialized organs which have only half the number of chromosomes and half the amount of DNA as compared to the non-reproductive body cells. Thus, when these germ cells from two individuals combine during sexual reproduction to form a new individual, it results in re-establishment of the number of chromosomes and the DNA content in the new generation. If the zygote is to grow and develop into an organism which has highly specialized tissue and organs, then it has to have significant store of energy for doing this. In very simple organisms, it is seen that the two germ cells have not very different from one another or may even be similar, but as the body design becomes more complex, the germ cells also specializes. One germ cell is large and contains the food stores, while the other is similar and likely to be motile. Conventionally, the motile germ cells is called the male gamete and the germ cell containing the store food is called the female gamete. We shall see in the next few sections how the need to create these two different types of gametes give rise to differences in the male and female reproductive organs, and in some cases different in the bodies of the male and female organisms. Sexual reproduction in flowering plants. The reproductive part of an angiosperms are located in the flower. You have already studied the different parts of flowers: sepals, petals, stamen and carpels. Stamens and carpels are the reproductive parts of the flower which contains the germ cells. What possible function could be petals and sepals serve? The flower may be unisexual, for example papaya, watermelon, when it contains either stamen or carpel, or bisexual, hibiscus, mustard, when it contains both stamen and carpels. Stamen is the male reproductive part and it produces pollen grains that are yellowish in color. You must have seen this yellowish powder that often sticks to our hands if we touch the stamen of a flower. Carpel is present in the center of flower and is the female reproductive part. It is made of three parts. The solemn bottom part is the ovary, middle elongated part is the style, and the terminal part, which may be sticky, is the stigma. The ovary contains ovules and each ovule has one egg cell. The male germ cell produced by pollen grain fuses with the female gamete present in the ovule. This fusion of the germ cell or fertilization gives us the zygote, which is capable of growing into new plant. Thus, the pollen needs to be transferred from the stamen to the stigma. If this transfer of pollen occurs in the same flower, it is referred to as self-pollination. On the other hand, if the pollen is transferred from one flower to another, it is known as cross-pollination. This transfer of pollen from one flower to another is achieved by agents like wind, water, or animals. After the pollen lands on a suitable stigma, it has to reach the female germ cells which are in the ovary. For this, a tube grows out of the pollen grain and travels through the style to reach the ovary. After fertilization, the zygote divides several times to form an embryo within the ovule. The ovule develops a tough coat and is gradually converted into seed. The ovary grows rapidly and ripens to form a fruit. Meanwhile, the petals, sepals, stamen, style, and stigma may shrivel and fall off. Have you ever observed any flower part still persisting in the fruit? Try to work out the advantages of seed formation for the plant. The seed contains the future plant or embryo which develops into seedlings under appropriate conditions. The process is known as germination. Here one seed is shown, germination. Now activity 8.7 is shown. Soak a few seeds of Bengal gram, that is chana, and keep them overnight. Drain the excess water and cover the seeds with a wet cloth and leave them for a day. Make sure that the seeds do not become dry. Cut open the seeds carefully and observe the different parts. Compare your observation with figure 8.9 and see if you can identify all the parts. Germination is shown here: a radicle, plumule, cotyledons in which food is stored. So here we will stop and we will see remaining part of chapter in next lesson, reading in next lesson, because to upload this full file in one attempt I think it will become difficult. So this next part, reproduction in human beings, we will see in next video lecture, reading of this, and then we will start discussing this chapter with the help of video lecture and PowerPoint presentation. Thank you very much.